Hello everyone. Now let's start with another topic in this session, that is about your RNA. Yes, about the ribonucleic acid and what is the role of RNA in the life processes and how actually this RNA is obtained from the most important thing, that is your genetic material. Now see, in our body we know we have the genetic material as the DNA, deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. Now, what is this difference between the deoxyribonucleic acid, the ribonucleic acid? Now, see, structurally we know that when ribonucleic acid, it has the ribose sugar. In the ribose sugar, especially at the, you can say, the second carbon atom, the particular hydroxyl group loses its oxygen, as a result of which the ribose sugar becomes deoxyribose sugar. Okay, and that's where this deoxyribose sugar started getting replaced in the ribonucleic acid and it became the DNA, that is the nuclear, you can say the genetic material of our body, of our cell. Now, talking about how do we obtain the RNA. When we talk about DNA, you can say they constituted genes, no doubt in that. But these genes, the DNA, it has to be expressed in our body. How it will be expressed? The first and basic question. If DNA is nothing but your genes, genes have to be coded. They have to be coded in the terms in which they can be expressed. Until and unless a gene is able to express itself, you can say it is just absolutely silent. It is just sitting as such in your body. So how do genes express themselves? How is it that a particular characteristic which is being governed by a gene is able to express itself completely in the body? So the first and foremost thing that we need to understand before we proceed into this RNA, this so-called DNA, nuclear material and all, is something called the central dogma. Now, what does the central dogma say? Central dogma say that whatever nuclear material you have, it has to express itself, and the sequence of events that helps a particular nuclear material to be expressed as such in the whole of your body, in the form of any morphological attribute, in form of any functional attribute, that is called the central dogma, and it involves, as I said, multiple steps. So what are they? Let us see. The first step will be the synthesis of more amount of your nuclear material, first and foremost, and in case of our body it is nothing but the DNA replication, or you can say simply the replication. In case of our body, I am saying DNA because DNA is the nuclear material in our cells. After this replication process in which the DNA is being synthesized in more quantity, you can say the content is totally doubled. Now talking about what happens next, more of DNA synthesized, fine. Next is the transcription. Fine. Now what is transcription? Transcription is the process in which we have a machinery which is able to synthesize RNA, or you can say messenger RNAs, or you can even say the various nuclear RNA or the mitochondrial RNA. So these all things can be generated out of the single DNA. Yes, it can be generated out of DNA, and what helps us to obtain this? It is the enzyme machinery, the various factors, the coenzymes, the coactivators, activators, mediators, and there are a lot of chemical components in our body, inside the cell, in fact I should say, which are able to completely transform or able to utilize the DNA to extract out the RNA. So you can say the first and foremost that if the DNA has to be first expressed, the first thing that we require is the RNA, that is the ribonucleic acid. Fine. Next, what? What is the next step in order to get a particular gene expressed? That is translation. Translate means from one form, it has to be modified into another form which is able to express itself. In the basic terms also, you can say about the language which we use, the English language, we also use various modes to translate one language to another. So why is it done? In order to understand that particular thing is being said in a particular language which we are completely lost about, or we have no idea regarding that. So similarly, the RNA over here, it has to be translated, no doubt, and translated into what? One language into another. Over here, RNA will be translated to what? It will be translated into proteins. And you can say proteins are those biomolecules which are actually able to express the particular characteristic which is being governed by that particular gene. For example, I have inherited the genes of the hair structure from my father, for example. So until and unless those genes have expressed themselves in the form of some keratin protein or the factors which are controlling the protein binding or the protein folding structures and all that, until and unless they are expressed, I won't be able to show off that this is my hair. I won't be able to see myself having hair also if those things are not expressed. So any body feature, any morphological attribute which is governed by a gene has to be expressed in its own form until and only by, you should say, only by the way of this central dogma, that is first replication, doubling the DNA content, transcription means transcribe it into another form, that is the RNA, and then the RNA has to be translated to the proteins which are actually able to express themselves. Now question is, what helps us to carry out the central dogma? No doubt, central dogma requires, in order to be carried out smoothly, it requires the smooth functioning of the machinery. Which machinery? It is the enzyme machinery. Means, what does this mean? It means that a lot of enzymes are required in order to carry out the central dogma. So I hope I have made myself a bit clear. Now question is, what exactly is DNA replication? So today we will be actually following up with DNA replication. We will see that what is DNA replication, how is it different in prokaryotes, how is it different in the eukaryotes, and where exactly do you see the differences in some cell cycle stages in various types of cells, and how it is going to affect the quantity of DNA that has been synthesized. And simultaneously we will be having a look over the various models of the replication which has been given to us by some great scientists, and simultaneously we will be seeing the type of replication or the model of replication in various types of cells. For example, the prokaryotic cell, in eukaryotic cell it is completely different. Then talking about the, you can say, bacteria, you can talk about the plasmid DNA. So these are the things which are going to show you some sort of alterations or some type of differences in the models that are able to tell you that how different it is, how each and everything is different from the rest. So let's move on to the DNA replication, and first, before I to another model that was actually expressed, or you can say the evidence expressed by the scientists for eukaryotic cell exhibiting semi-conservative mode of, or you can say semi-conservative model of replication. And when we talk about prokaryotes, be careful, it is the organism that was used to express this semi-conservative model of replication. It was the E. coli. And when we talk about eukaryotes, these are the broad beans, or you can say the scientific name was Visia faba. So these are actually the broad beans, you can say the plant cells which were used as the model cells to exhibit the semi-conservative model of DNA replication, especially with reference to your eukaryotic cell. Now talking about who gave this experimental evidence, it was actually Herbert Taylor, a combination of three people. Herbert Taylor, second person Philip Woods, Herbert Taylor, Philip Woods and Walter Hughes. Walter Hughes. So these were the three people who actually gave the experimental evidence with reference to the semi-conservative model of replication in eukaryotic cell of Visia fiber. Now talking about a bit of another thing or another model, you can say, that is about something called the semi-discontinuous model. Okay, so this is actually a big hint. Okay, let me just explain it a bit to you. Why is it called the semi-discontinuous model? Because whenever DNA is being synthesized during the replication of DNA, it is synthesized in two ways. Okay, you know that there are two strands antiparallel. They will be separated at one point. The base pairing, hydrogen bonding that is present, it will be separated for a while, and during this separation, for example, if this is a portion where they are being synthesized, new strands are being synthesized, let me tell you, if this is the 5' end, this is the 3' end. If this is the 3', this is the 5' end. Normally in our DNA double helical structure. So whenever a new strand is being synthesized, it is always in the direction of 5' to 3' direction. It is always in 5' to 3' direction. Now what is this 5' and 3'? We will discuss that. But this is a continuous format. For one strand, the complementary strand will be synthesized in a very continuous manner. Means if it starts from here, it will go on being synthesized. The nucleotides will be added completely up to the entire length. Not an issue. But when we talk about the other strand, it is not continuous. You can see I have drawn some breaks. Why? This strand in particular is synthesized in fragments, in portions. And what are these fragments called? These fragments form the, you can say, the lagging strand. And this is called actually the leading strand. Why it is the leading strand? Because it is highly continuous, no doubt in that. We can see it clearly. And when we talk about the other strand, that is the lagging strand, they keep on lagging behind. Means they get constructed in fragments in short intervals. And these fragments have been actually named after the person who gave the semi-discontinuous model of replication. And the name of the person who gave this model was Renji Okazaki. And that's why these fragments are called Okazaki fragments. These fragments are called the Okazaki fragments. That's it. So that's all about your semi-discontinuous model of DNA replication. And no doubt in this that the DNA synthesis or the nucleotide synthesis always takes place in one direction, that is this 5' to 3'. But when we talk about the leading strand, if this goes from top to bottom in one direction, the lagging strand synthesis is also in the same direction, that is 5' to 3', but it goes in the reverse, you can see. Why is this happening? Because these two parent strands, they are not parallel, they are anti-parallel. Means if this is 5' and 3' for here, the other strand, it will be just anti-parallel, means upside down. 3' will be top, 5' will be bottom. That's the reason why we can see the leading and lagging strand being continuously, or you can say discontinuously in case of lagging strand, being constructed in opposite directions. Okay, fine. Now let's talk about some more things, like especially the enzyme machinery that controls the DNA replication in prokaryotes. Okay, let's have a look at the enzyme machinery that controls the DNA replication in prokaryotes. So the enzyme machinery, the first and foremost thing that actually we can talk about is the DNA A protein. Now question is, why I have written this in small letters? Why not in capital? Now in small letters, because these are the proteins, or these are the enzymes, you can say, which are playing a specific role in the you can say, activity of DNA replication. So that is the sole reason why I have mentioned it as such. Now talking about the function of the DNA A protein, it actually acts like the enzyme which is responsible for the localized strand separation. Very important, localized strand separation. Now question again, that localized means it is occurring at one particular place. So which is that particular place? It is actually the origin of replication. It is the origin of replication, which we call, in case of your E. coli cells or the prokaryotic replication, ORI-C. The point where the DNA replication is being initiated, it is the ORI-C, or for which the DNA A is responsible, especially for localized strand separation. Until and unless both the strands are separated, the replication procedure is not going to be carried forward at any cost. Now talking about DNA C protein, okay. The DNA C protein is actually responsible for creating a bidirectional fork. Now this is very interesting thing, something which I had drawn just a few seconds ago. For example, if this is one end, this is the other, anti-parallel strands of the DNA. So this is actually the replication fork that is being created. It's a bubble, you can say, and if you just examine this particular portion, then it acts as a replication fork. And why it is bidirectional? Because if the lagging strand is being synthesized in this direction, the leading strand is being synthesized in this direction. That's why this bidirectional fork that is actually created by the enzyme DNA C. So actually these are very important when we talk about this in terms of your objective questions. They just give you an MCQ where the alternatives will be with reference to the various enzymes, and they might have given you the question as a particular activity where you have to identify the exact enzyme which is responsible to carry out that particular activity. Now talking about DNA helicase. Now helicase has been actually taken from the term helix. Okay, so let's talk about DNA helicase. DNA helicase is actually responsible in introducing negative coiling. Negative coiling means DNA is already coiled. It will start uncoiling it, which I call the negative coiling. So if you uncoil it, what is going to happen? It is going to get opened at one particular juncture. At one particular juncture, it is going to get open. This is the activity of this DNA helicase. Now you can see over here that if this DNA helicase has been successfully able to separate the two strands of this DNA, then don't you think that there are chances that they might be again coiled back? Yes, that is possible. So over here another set of enzyme comes and binds over here to these two strands, parent strands, which are called the SSB proteins or the single strand binding protein. Single strand binding protein has the most important role of maintaining the stability of this single strand. Because why I'm saying, for example, you know Chargaff's rule, in which adenine always pairs with thymine and guanine pairs with cytosine. Now question is, if in this sequence you can see T, C, G, G, C, A, T. Now there are chances that there might be a coiling or a kink, appearance of a kink in this particular parent strand, which might result in the pairing something like this. So I hope you can see this. So this is possible, T, A, T, and these are called your hairpin structures. These hairpin structures are strong enough to stop the replication machinery over there itself. So in order to avoid all these things, the SSB proteins play a very important role of maintaining the stability in the single strands when formed by the DNA helicase. So I hope this part is clear. Now talking about the most important thing, that is your DNA topoisomerase. Now topoisomerase actually has two types. One is DNA topoisomerase 1, DNA topoisomerase 2. Okay. Now what is the function of topoisomerase 1 and topoisomerase 2? Now the first and foremost difference between the two is this is ATP independent in its activity. It doesn't require ATP. This is ATP dependent, means it requires ATP in order to proceed forward. Now talking about the similarity, yes, both of them, or you can say topoisomerase 1 and topoisomerase 2, they have nuclease as well as ligase activity. Means nuclease means they can create cuts in the strands, and ligase means they can seal the strands, or you can say seal the cuts in the strands. So this is the benefit, or you can say a sort of similarity between DNA topoisomerase type 1 and type 2. Now in case of E. coli, one type of DNA topoisomerase 2 that we find is DNA gyrase. So many a times there is a confusion amongst the students that what is the function of DNA gyrase, or what is the similarity between DNA gyrase and DNA type 2 topoisomerase. So this is actually, you can say, the difference, that DNA type 1 topoisomerase, it is responsible for producing the same sort of effects in case of as compared to DNA topoisomerase 2, but it is ATP independent in comparison to type 2, which is ATP dependent. Now both of them, they have this nuclease as well as ligase activity. Means nuclease means it can produce cuts, it can produce kinks in the DNA, whereas in case of ligase means it is able to seal the thing completely. Means if there are any cuts, then it will completely seal it. That is the benefit of the ligase activity of the DNA topoisomerase 2, and this thing is exhibited by DNA gyrase. Now question for you is DNA gyrase ATP dependent or ATP independent? As we said that it is a type 2 topoisomerase, so it is ATP dependent. This enzyme requires ATP or investment of ATP in order to exhibit either of its effects. I hope this thing is pretty clear. Now talking about RNA primers. Now RNA primers, this thing is very important. Why? Let me tell you, DNA is the genetic material, no doubt, strong enough, fine, but when we talk about replication, means synthesis of new nucleotides and they being continuously joined with the help of your phosphodized linkage, the initiation is not possible by the DNA itself. It always requires the help of RNA. And how is RNA present? It is present in the form of RNA primer. The RNA primer has a, suppose this is the 5' end, this is the 3' end. It always has a hydroxyl group attached to it. The 3' hydroxyl end, it is always open, to which you can say the new nucleotides will come and go on binding. Now the question, as I had said, that I'll explain what is this 5' and what is this 3' end. Let me just tell you. Suppose this is your sugar, pentose sugar, talking about deoxyribose. For example, deoxyribose. So you can see these are the 3 phosphate groups. This is the fifth carbon atom. This is the 1, 2, 3, 4, 5. This is the 3rd carbon atom. So it goes in the anti-clockwise direction, the numbering. Now from this 3rd carbon atom, this thing will be joined to the 5th carbon atom of another, you can say, sugar. This is it. This is how the attachment goes on in case of any nucleotide sequencing. So that's why we say it is always synthesized in 5' to 3' direction. This is the reason. The 5' is the 5th carbon atom, 3' is the 3rd carbon atom. So I hope this thing is pretty clear to each and everyone who is going through it. And RNA primer is very significant because the 3'OH end of the RNA primer is responsible to where all the nucleotide sequences will go on being attached with the help of phosphodiester linkages. Now, what helps? Where does this RNA primer come from? Now this question, where does this RNA primer come from? RNA primers, they are actually synthesized. They are synthesized. How? Now that's also, now there are more questions, there are more enzymes for this. One is primase, the primase enzyme which is helping in synthesis of RNA primers, and another one is your RNA polymerase. Since it's an RNA, since DNA has its own polymerase where it can polymerize the synthesis of your nucleotides for making up the DNA stack. So similarly, you can say that RNA also has an RNA polymerase. So it's the RNA polymerase and the primase which are actually helping in creating this primer and exhibiting its 3'OH end vulnerable enough to accept all the nucleotides that are being synthesized by the DNA polymerase. So this is how it works. Now, most important thing is the RNA primer going to persist there forever during the DNA replication? Actually, no. It will be removed. How? We will see that. But before seeing this, we need to first see DNA polymerase and DNA ligase. Now see, DNA polymerase definition is simple. How? The DNA polymerase is actually the enzyme which is polymerizing this reaction of adding more number of nucleotides so that the DNA strand can be synthesized, complementary to the parent strand. So the polymerase, you can say it is of 2 types in case of your prokaryotes. That is DNA polymerase 1, 2 and 3. Now I am not going to just go on blabbering about DNA polymerase 1 is like this and that and 2 is like this and that. No, let's go for the comparative analysis. Means comparatively we will see that what are the effects of this polymerase 1, which things they can do, which things they cannot, so that it becomes very easy for you to understand, and technically also if any question is being asked regarding this, you are able to pick up that what exactly should be the answer. Now for example, what are we going to judge polymerase 1, 2 and 3? On what grounds? You can say one is the structural polymerization, means they are able to synthesize the nucleotides or not. On the basis of that, yes, all 3 are capable of doing that. Another one is exonuclease activity, means cutting the nucleotides, removing the nucleotides, that is exonuclease. And that too removing the nucleotides from end. If any enzyme is removing nucleotides from ends, it is called exonuclease. If it is removing nucleotides from the middle, they are called endonucleases. So exonuclease activity in 3' to 5' direction, exonuclease activity in 5' to 3' direction. So these are the 3 criteria in which we will be seeing that what exactly the functions are of polymerase 1, 2 and 3. Now exonuclease in case of 3' to 5' direction, yes, all three are capable of. But when we talk about exonuclease activity in 5' to 3' direction, only one is strongly capable enough. DNA polymerase 1. And if you go on talking about DNA synthesis, proofreading, excision of RNA primers, then it is polymerase 1. So we can write it down as, yes, it is capable enough of proofreading, synthesizing new fragments, synthesis. Then you go on with, you can say polymerization, or you can say the synthesis, proofreading, and yes, excision of some nucleotide sequences in 5' to 3' direction. If I talk about DNA repair, it is only polymerase 2. If I talk about DNA repair, it is polymerase 2. And if I talk about proofreading of newly synthesized DNA, so you can say proofreading activity in a way is carried out by both 1 and 3. But 1 is the most active ingredient, 2 responsible for DNA repair. No doubt in that. So you can say this is the comparative analysis of DNA polymerase 1, 2 and 3. And polymerase 1 was actually discovered by Arthur Kornberg, and that's the reason why this is known as the Kornberg enzyme. Very important. Many a times this question comes as objective. Now, as I said, DNA polymerase 1 is capable enough in excision of nucleotide sequences, that is in 5' to 3' direction. Now, this is something important. Now, let's see how. Now, as I said, the RNA primer is present. This is the 5' end, goes on attaching the nucleotides. Now, for example, this is the leading strand, and we talk about lagging strand. This is the lagging strand. Now, is this possible that the RNA primer stays as such forever? Definitely not. It has to be removed. How it will be removed? Over here plays a very important role by the DNA polymerase 1. How? We said, or we studied, that it has 5' to 3' exonuclease activity. Means from here RNA primer will be removed. In case of this, fine, it's okay. No issues in case of this leading strand, not a problem. Problem will be faced by this lagging strand. Why? There are gaps. Even if they are synthesized, they stay as such because RNA polymer has been removed. And if RNA primer has been removed, what will happen to these gaps? These gaps will be sealed. By whom? DNA ligase. So you can say that this activity is the result of the dual role played by the DNA polymerase 1 as well as DNA ligase. Without these two, it wouldn't have been possible. So DNA polymerase 1 is responsible for excision of nucleotides in the 5' to 3' direction. No other polymerase is able to do that. But this gap will be sealed, no doubt, with the help of DNA ligase. So this is how the lagging strand is also made or converted into a continuous one. So I hope this enzyme machinery about prokaryotic DNA replication is clear. In the next coming session, we will see about the enzyme machinery of enzyme machinery of eukaryotic DNA replication, and apart from that we will be covering the prokaryotic replication processes and the eukaryotic replication processes. So till then, stay tuned. If you have any questions, you can type them in the comment box, or if you want, you can just mail me at the mail ID being provided from here on.